Yeah, and then you should also have, I enclosed 10 or 2 packs of 5 glass slides that you should see. And in here, I've included cover glasses that are wrapped up in this tissue paper. So when we work with these, just have to be careful with them because they're very small, very thin, and they break extremely easily. So, and you should also have a pipetter with you. And if you had some of the pipetters from the culturing exercise that aren't too dirty, you can utilize those as well. Because with the microscopy, we're not so concerned in this context, we're not so concerned about sterility. Because as long as they're well rinsed, you probably won't see so many structures, maybe with the exception of the yeast, but we're not going to do it that way. We're going to do it in a way that you won't see yeast cells all over the place with every slide that you make. Okay, but in addition, hopefully you've been able to get a few of the extras. So you should have a pair of tweezers, pincette. They can be any kind, metal, whichever, but that'll work. Yep. The toothpicks are very handy for like manipulating the sample on the slide, on the cover glass. If you need to, for example, with the hyphae, you can spread apart the hyphae on the slide so you can visualize it better. Hyphae can be difficult because it clumps up and makes a clump that you can't really make out any structures on a slide sometimes. But if you have the toothpicks, you can spread it out a little bit and then put the slide on the cover glass, and it might make it a bit easier to see hyphae. Scotch tape. And again, with the dry yeast, but you should have all that set. You should also, with your kit though, do you have an extra magnetic coupler that can go on the phone? No, I see some heads shaking yes and some no. Uh-oh. Oh no. Meh. Ellen, you don't have one.
No, I didn't receive one. I saw in a YouTube video when I assembled it yesterday.
What?
But I didn't get one.
I couldn't find one either.
You're missing one.
I think I only got 2 for this and then the lens. Is that correct?
Yeah, but there should have been an extra one in there.
No, no extra.
And 3 that was coupled together.
Yeah, they were quite kind of difficult taking apart.
Yeah, because really there were 3 of them, right?
Yeah.
For me, it was.
Double check.
I had 3, and they're big. You had to pull them. It felt like I was gonna break them, but they didn't break.
Exactly. No, double check because they might have been stuck together. Everyone should have had 3, because there were 2 that go on the scope. So one with the lens and one for the back, and that clicks. And then there's another one that goes on the top like that. And that's the one you put on your camera. And then I included an extra one for the back of the illuminator.
Oh, I know what I did.
Ah.
Because the lens was stuck between 2.
Yeah.
So I kind of assumed that the lens was actually on that rectangle too. Oh. But it wasn't. It's right here.
Yeah. This is why we're going through it. I'll give you a little bit of time to fix it. Yep, there it is. Good.
So where was it? There's a third one.
One of the other ones. I thought that the lens came on one of the plates too, so I didn't see that it was a lens and one of the magnetic holders.
Ah.
So when I separated them, I clearly saw that the lens came just as a round thing.
Yep, the lens is just a round piece, not a square piece. The lens detaches from that.
Is this the one you're talking about?
Yeah.
Okay, okay.
Because that should be the lens, right? Because it looks like you have the lens in there.
Yeah, it isn't. Okay.
because the ring itself is just this. There's no lens in it. It's just a metal ring with a black piece of plastic on the outside.
Where did this go?
Give me a second. Let me take one out. I'll show you. I got another kit here. So when you receive the kit, I think you had this.
Yeah.
So when you open it up, you might see that there's one, and that's just a ring.
Mm-hmm.
Two. So this is just a ring as well. This is the lens. You can see
Okay.
that the lens, if you look on the back side of it, if we take it off like that, this is the lens. And this is another black ring.
Oh, okay. I see now. Okay. Ah. I see.
So then, when you've assembled it, this is, if you look in the assembled one, so I can do like this. Oh, I kind of, there's an adhesive ring in there, so I can't really take it out.
So, okay,
But-
and now it's 3 pieces and I have this one.
Right.
Okay.
So if you look at my assembled one, here's the lens again. This is the back side of the lens.
Yeah.
So to compare it, that's how it sits in the microscope. And if we flip it over, you see something like that. But this is the top part of that ring. So in essence, you have that,
Okay.
right?
Yeah.
And I think they had you put adhesive rings so that the lens doesn't come out. Yeah?
Yeah.
Do you need to fix anything or?
I think it should be. Wait, should it look like this? Just.
Yeah, yep.
Okay.
This is why we're going to test it with a slide that I know works.
I think I have some problems, so I should separate this. Where's the camera?
Let me see.
Yeah, that's the lens.
Yeah,
Yeah.
exactly. Yeah, because you should take.
I'm
you
gonna
should be
need
able to
some-
take. Because the lens should be taken out when you're building the scope, and then like one of the last steps is that you put it in with an adhesive ring, if I remember
Right.
correctly.
So if I just separate these two. Oh yeah.
Yeah,
Yeah, perfect.
yeah.
Thank you.
Yeah, so now I have an extra one.
Yeah,
We can.
good. Because that one can go on your camera for your phone. Oh, I'm laughing because I didn't know how this was going to work out digitally. It's
So
always
where's
so
the-
much easier. We can go from person to person in the same room and point out things and play with things.
Chris, I
Yeah.
forgot where to put the lens. Where should I put it?
The instructions out again. Right. So the lens goes, let me see here, just to double check. Do you have the instructions with you?
Yeah.
Yeah, because if I recall correctly, so this is the back side, right?
Yeah.
If you open it up,
Yep.
the lens should go on the back here, right?
Yeah.
Because if you've inserted this piece into this top part here,
Yes. So yeah, and it's magnetic, so
right, exactly. But there's I think you have to use they want you to use a piece of adhesive tape. There's a clear piece of adhesion that you want to put in there too.
all right.
It's the
Look
small
at,
white ones.
yeah, the small white ones.
The white ones. If you check Chris's left Left corner there, you can see the white ones. Yes,
Yeah.
they're double-sided adhesives.
Double check in the instructions, double check step 4. Yeah, exactly. Right. So you'll see that you'll have that black piece in there. You put, here, you know what? I'm just going to take it out. So if it looks like this to start with, there's no lens in here now. You'll have the lens like that. And they want you to put like a sticker here. One of these guys. So that gets put right onto that circle, okay? And then you take the lens, you flip it over, and it goes right onto that circle, okay? Then you should have the lens in place. The adhesive is important because this back part is magnetic, and it's going to click on here like that. And that's what's holding the slide in the microscope, but you don't want the lens to move when you're adjusting the slide, when you're adjusting the microscope and the focus. This slide here is what we're going to use to test that it's working correctly. So what I'll do is I'll just show you how we insert the slide and then view the specimen. So like I said in the instructions, there's a picture of what you should see if your microscope is working properly. It's these little silica structures, these diatoms. So just get everyone's attention real quick here. So if you look at my hands, so here I have my scope. To insert the slide, what we're going to do is we open up the back like that. Here's my slide. This is that's the top part of the slide, so the cover glass. So if I have it like this, I'm going to flip it so that the cover glass is facing the back of the lens, okay? Now this is a little bit tricky, but you see these tabs here, like that? You're going to take the slide and it goes under the tab. And it takes a bit of wrangling. You got to really work with it. But the slide should go under the tab like this. If you can see how that is. So there's this little piece sticking up, and the slide is just under that, but still inside the back piece. Maybe you can see that. Yeah? Then you have to slide it in a little bit and then put it on the other, underneath the other tab. So like this. In the end, you should have something that looks like that. So here I have my slide. You can see it reflecting the light. And it's being held in place by this part and this part on, well, this is what we will call the stage on a microscope. So this is what is holding the slide in place that we can then move later. I'll leave this up so you can see what I mean if you get a little lost. So then once you have it in there, all you have to do is click the back part on like that. Take your illuminator, put it on the lowest setting, on the lower setting. Click it on the back. Now, you should be ready to view when you have your illuminator and the slide in place. Now let me go through the different parts here, because this is acting as kind of a microscope, like a stage. So we have different aspects of that, meaning that we have movement in all three planes. So we have this X movement here, so we can go back and forth. It's a little bit tricky because it gets a bit stuck, but that's because of the illuminator. You can go back and forth in this direction. You can go up and down in that direction. This is the focus. You see how it's like a wedge?
Oh, I didn't stop. Well,
What's up?
oh, it's nothing. I just realized my mic was on. It's fascinating. Just
Okay. So I'll just continue. This is the focus, right? So as you push in the wedge, what it's doing is it's raising the lens away from the slide. So you're going up and down in what's called the Z plane. That gives you the focus. So what we'll do just to make sure is make sure that the slide is centered. It's a little bit wonky, but you can, after a little bit of playing with it, you can get it. Make sure the slide is centered. Our light back on. And then I'm going to take a look like that. You might have to adjust the light. I'm going to do something like this first, but give me a second here, because the easier thing is going to be for me to put this on my camera and I can show you. I was going to get to that step next.
I've succeeded to get a photo already. I don't know if you can see through here.
Yeah, there you go.
Look at that. Works.
Perfect. You got it. You can actually zoom in a little bit. That's actually, so did you put the magnetic coupler on? Yeah, because that's what I'm doing here. Because with an iPhone, it's annoying because I got the camera's on a bump. So I have to use a little extra tape. But let me just, I need to do
That's why you don't have an iPhone.
Yeah, that should be the general rule. Don't have an iPhone.
This is kind of insane. Crazy good. I mean, I have looked through hours in microscopes at the hospital, but this is like crystal clear.
Not bad, huh?
It's super good.
Yeah, I was shocked when I first started playing with it, but it's pretty good.
Where did you put the coupler for the phone?
I'll show you. Because of the iPhone, I have to do something a little bit special. What I've done is I have a piece of tape like that, and then I put the coupler on the tape, but I cut a hole using tweezers or a little knife or whatever, and I cut a hole, and then if you see my phone here, I take it and then I center the ring so that it's over the very center part of the camera. It can be a bit tricky, but after some playing, I'm able to center the ring well enough where you should be able to see that. Focus is a bit bad on this camera. And because of the iPhone, I need to put another piece of tape to make sure it stays secure. And I do that by putting it down here underneath. And that seems to work for me. I think those of you that have Androids or other types of phones, you probably have an easier time, right, Malin?
Mine is not magnetic enough, so I have to tape too.
Not magnetic enough. It's not sticking?
It's not sticking. But why don't you just use 2 pieces of tape so you don't have to do a hole?
Whichever works. For me, I use it like this, and then it stays in place quite well. So I think everyone just has to sort of play with it until they get it working satisfactorily. So now that I have my phone, I think what I'll do is like this. You. There we go. And then if I'm able to put the, give me a second, let me just adjust the camera here. Coming back to our cooking show again. So I put it on square, and then you should be able to connect it with a magnet like that. And there we should have an image. And I can see the little structures. So it's actually not a bad picture at all. And maybe you can see how I'm doing it. I can leave it in place and then just focus it using the wedge and draw it back and forth. And then eventually I can get it in focus. You might have to adjust the light, just push it underneath if it's going out of center. And there we have it. So I will take a picture as I have it now. And probably can't see it so well. I have an example up in the photo gallery. But you should be able to see clear, distinct diatom structures. So small little squares, pieces that have been broken off, but then they should be quite distinct. How's everyone doing?
Chris,
Yeah,
can you please repeat where I should put this?
right. Let's go back to that. So,
Thank you.
no worries. So what we're going to do, let me just take it out and I'll demonstrate one more time. So here we have our microscope, right? So what you do is you're going to open up the back part like that. So here we have the slide, and I'm holding it so that the cover glass is up, okay? So you should feel the cover glass, and it should be able to see, you should be able to read the letters DE test. So I'm going to flip it over so that the cover glass is facing the lens. Let me just turn a little bit. The slide fits in under these 2 tabs. So here and here. So if I take the slide and then I go under the tab, and you have to wedge it a little bit, but you just work it in until you have it all the way in. And then do the same for the other side. So you just bend it around a little bit and push it in. And then you put it back into center. Now you have the slide mounted in your stage of the scope. And then all you have to do is click the magnet back on, and it should clamp on to the slide. You able to get it? Yeah, I know it's probably a little difficult the first time, but after a little bit, it'll go. Yay! I love a thumbs up. Okay, so once you have it, you can take your illuminator, right? Make sure that the switch is so that it's on the low light setting. And then all you have to do is clip it to the back. And to start with, what you can do, what you can do is hold it up and you can look in here and you should be able to see small little structures. You might have to move the light a little bit because sometimes it can be a bit off center. So you just push it back and forth with your fingers. But remember, this part here, this is the focus. To focus, what you have to do is you move this in and out. That moves the lens up and down the Z plane. So as you adjust that, you should start to see some structures coming into resolution. You see anything? Good. Yay, then it works. Everyone's on board. Now everyone has a fully functioning microscope. Is it okay? Do you have it going, Fanny? Yep. All right.
I have a question. I managed once to get a picture, but then every time I put my camera onto the scope, I only get a little a quarter of moon in one corner, but I can't focus it. Even how I try to move the camera and I try to move the lights and I don't know really what. So I can't see anything there.
Can you show me what it looks like real quick? Yeah, you know what happens there? If this ring, oh God, it's working upside down. I'm gonna have a massive headache by the time I'm done with this. If this ring is not perfectly centered on the camera lens, it'll make that half moon and then you can't adjust it. You really have to make sure that the very center of this ring and the inner part is really centered on the lens of the camera and the actual iris of the camera. That's the tricky part. Because then you won't get that half moon.
Okay, I'll try that.
Yeah. I struggled with that quite a bit when I started playing with these. That's why for me, that's why I have to use so much Scotch tape. Because then it makes it easier for me to center it. Because you can see here, I have it fairly centered and it's just the light that's not centered. I can move the light and then it changes it. Okay, if you just take a look here, this is the focus, right? So as you slide this ramp in and out, that's what's bringing the plane up and down on the lens. What can be a little bit tricky is to try to keep everything in place when you're working with it. So what I like to do is when I have the the light on the back, and then I'll keep my fingers there and just try to hold it in place. And then I can use my thumb and then just sort of bring it in and out while keeping the light completely still in the back. So you can probably see how I'm holding it like this. That helps a little bit to keep it steady. But that's just how I'm working with my hands. Everyone has different like types of dexterity and how they get comfortable with working with this. So whatever works for you, but this is how I'm doing it. Because then you should be able to look through and then see it coming in and out of focus as you're going back and forth with that ramp. So what we'll need is we'll need our cover glasses. And I'm going to take the slides out here. I have my slides there. I'm going to take the cover glasses out as well. So I have, there should be about 20, probably 30 cover slips in this little folded piece of paper. So if I ever so delicately open it up. Oh, by the way, what I like to do whenever I'm preparing slides is I put down a paper towel on the table. This is just preference, but it kind of helps me keep track of things. And it also, if I need to wipe something up or blot something, I can use a little bit of the towel. I'll show you what I mean in a minute. But I'm going to unfold this little piece of paper here and get at the cover slips. So you see them right here? So you can see how small they are, and they're incredibly thin. And they break quite easily, which also means that you can get glass shards quite easily. So handle them quite carefully. And if they do break or if they crack or whatever, just use a paper towel and kind of wipe up the glass. But always handle them, by the way, whenever you pick one up, always handle it by the corners. You know, don't put your fingers on over the slides, obviously, because you're going to dirty up the cover slip and then you won't be able to see very much. So whenever we handle a cover slip, you always pick it up by the corners and by the edges. Put it back down like that. So what I thought we could do is start off by just preparing a simple yeast culture and look at the yeast under the scope. We'll try it like this. I want to try to use these rings because I found them quite useful. So I'm going to get my tweezers and I'm going to pull off a ring. Okay. And I'm going to put the ring on the center of the slide and then push it down gently here so that it adheres to the glass slide. So then you should have something like that. Now you'll notice that there's a thin piece of plastic over the top. You might need to push at it, but try to peel that off so that you're left with the second adhesive side that's exposed. There we go. It's a little difficult to lift, but once you lift it, it'll come off. So now there should be a slide with the adhesive ring on it, and then there should be the adhesive side exposed. So we'll get our water and our pipetter. And all you need is just a very small drop in the center. I mean small. You shouldn't, it's almost like you're wetting the center of the ring. It shouldn't be a huge mounding pile of water. It should just be enough where it's just over the very top of that ring, just barely over it. Okay? So for example, if you hold it up and look at it from the side, the drop should be more flat than not. In other words, don't use too much water, just enough. Now this is, if we have our yeast culture, if we were to just put this on the slide, we really wouldn't be able to make out very much. It wouldn't be able to see very much because it's too thick. There's too many cells. We really want to have a nice thin layer of cells so we can differentiate each of the cells. Otherwise, it just looks like a complete flood of circles and you can't make out anything. So what we're going to do is take a toothpick, open up the yeast culture, and then just dab the toothpick in, and then dab it around inside the water. Do that twice, and that should be enough yeast cells where you can see them. It really doesn't take much because there's a lot of yeast just on the edge of that toothpick. You can just put that to the side. Then we'll take our cover slip. So I'm holding it by the edge. I'll have my tweezers on hand just in case. And then I gently put the cover slip, you put it down flat on top of the drop, and then just push it right onto the ring. You might get a little extra water that flows out to the side, and that's okay. But then just take it and don't push too hard, but just sort of settle the cover slip on top of the ring. And it should stay in place. And if you have a little excess water, take a little piece of towel here and just blot the water. You see how it absorbs? Maybe not, it's a little bit hard to see. So you can just put it on the edge of the slide and then just suck away the water. That way you don't have a lot of water making a mess of your scope when you put it on into your stage in the fold scope. So in the end, you'll have something like that, where you have the ring with the yeast cells inside and the cover slip on top. Look okay? So that is preparing the slide. Now let's take a look at it. So we'll put it back in our scope. Underneath one tab, here, and then underneath the second one. Clamp it shut. Put the light, and now we should be able to visualize it. And with any luck, you'll see a whole bunch of small little circles, which are your yeast cells.
I see something.
Good. So if you want, you can take a picture or you can draw even because in the exercise, if you've printed it out, which not necessarily, but I've printed it out and you can draw on any piece of paper if you want. You can draw the yeast cells. That's kind of the classic way of doing microbiology or microscopy. Or you can take a picture using the magnetic clamp. So if I do that with my camera, and I zoom in a little bit here. There we go.
So
I'm
Chris,
gonna...
Hi.
Can you hear me?
Yes, I can hear you.
Yeah. So I'm almost getting into focus, but I want to pull it further apart, and I can't because it's a stop there. Have I done something wrong then? Should there be more space between the lens or something?
Pull it further apart. How do you mean? You mean you mean the ramp?
Yeah, the ramp. I want to pull it, like I'm almost in focus, but then it stops. So I want to pull it further out, but
I think if
it stops me there.
Okay, I see. I think you might be a little, so you mean, if you can look how my camera is, so you mean that you have it all the way out like that? Yeah. Let me see if I can zoom in a bit on my screen, because I think that's a bit off. You should be able to get it in focus, even because that's all the way out and it shouldn't have to go much further. Give me a second here. Let me get the light. Let me see if I can do this upside down.
I have it the other way around, so it's all the way, let's see, but the thickest in.
All the way in.
So like,
yeah,
like
that. Sometimes what you can do is simply flex the scope. If you look at what I'm doing on the camera, you might be able to see the screen, how I can change it. It's again fidgety, but you can focus as well with the lens just by adjusting, kind of playing and flexing the scope a little bit. Because if I was to leave this by itself, like not touching it, I can push in and then it starts to focus as I push the ramp in. Let me, were you able to see that on the camera? Give me a second here. I want to do like this. Whew.
I couldn't get any sharp pictures on my phone, but I think I realized that I've got a tiny scratch in the middle of the camera.
Oh no. Well,
but it's 4 years old, the phone, so I can't blame it, you know.
Yeah, the scratch will do it. So here, let me see if I can back out a little bit. There. All right, that's a better view. I just need to focus a bit and I can show you what I mean, maybe a bit better. So when I play with it, I'll hold it like this. You can see what happens when I just flex the microscope a bit. It'll go in and out of focus, but when I come out, yeah, you can probably see it's getting a bit blurry. There, that's better. And then when I start pushing it in, you'll see it's coming into resolution a bit better. But as I move it, just flexing it a bit, it goes in and out. So you might just have to sort of finagle it, meaning that adjust it how you're holding it. And that might help.
Vad har ni för förstoringar på era?
Vilken vad?
Vilka förstoringar ni har på... du sa att, vad sa du att det var på den här foldscope? Var det...
140?
140, ja.
140 magnification.
Och sen så har ju kameran också.
Yep. So then you're probably getting up into like, if you were able to do 2 times, you're probably getting into like 280.
Yeah.
Yep.
Got it.
You got it? Yes! Woohoo! I can show you a little trick too, if you play with the illuminator, because maybe if you remember from the lecture so many months ago, in microscopy, this is just a bright field microscope, right? So there's no contrast method. like the phase contrast or the DIC, a differential interference contrast, where you manipulate light in order to see cellular structures that are otherwise transparent. But with the light here, you can actually move the light a little to the side, and you can actually get, this is called dark field microscopy. So you're illuminating the specimens from the side. So the background is a bit dark. It's a little tricky to do, but the cells light up like little stars. That's another way in which you can get like that. You can get a little bit more resolution, or at least see structures a little better. This is just a little trick that I kind of figured out when I was playing with this. But dark field microscopy is also quite good if you want to see bacteria that might be swimming around at low magnifications. So that might be a little tip if you want to see bacterial cells later. Can anyone see any yeasts that might have little buds on them?
What do you mean by buds?
So when yeast divide, what they do is they produce tiny little bumps off of the side of the cell. They produce buds, basically, just small little cells that start to form and then grow bigger and bigger until they separate from the mother cell. And
do you mean like that one? Is this what it should look like?
A budding yeast cell, I can draw it out here. The way that yeast divide, if you can see the camera here, there we go. So what you'll have is a mother cell, but then sometimes you see a little bleb like that.
Oh, okay.
And then sometimes you'll see other ones that might look like that, like Mickey Mouse.
Yeah.
And then these are the daughter cells that are being formed off of the yeast cell. They might be kind of hard to spot because they are small under the microscope, but you might be able to see a small cell that has a duke like that on the top or on the side.
Okay.
See if you can find those.
Yeah.
I found a couple. Yeah, because if you didn't collect any hyphae, now you can just, I'm sure there's something moldy in everyone's fridge that you can look at. So go for it. I know that my fridge is definitely a nice large culture collection right now that I'm trying not to think about. So I'm going to make another slide. And we'll see what we can see for fungal hyphae. I've actually had the luxury of, like I said, I work in the Department of Forest Mycology, so I have access to all sorts of fungal cultures. This is actually Rhizoctonia solani. It's a pathogen of many different crops, actually, but this one is also a pathogen of tomato. So we'll see what that looks like.
So do you work with Anders Dahlberg?
I sit right next to him, yeah.
Ah, cool.
Yeah, he's very nice guy.
He was my supervisor for my bachelor's.
Ah, okay. Ah.
Yeah.
Yeah, he's actually been a very good resource for this course, for running a distance course like this. So I've been quite a lot chatting with him. Very nice guy. Have you taken the the mushroom course that he offers.
I've applied 5 years in a row, but there's like always 300 people applying for
Very
it.
popular.
So I'm taking the advanced course with him instead.
Okay.
I'm skipping it. Yeah.
Now it's a very popular course. It's a very nice course. So I have my black ring and the water drop here. And I have my fungal sample. Now that I'm making a note, I forgot where I put it. Come on, where did it go? Ah, right here in front of my face. So I'm going to take some toothpicks here. Actually, to make life easy for me, I'm just going to take the sample out here because I got a little bit of hyphae on a bit of auger here. So I'm going to take some tweezers and a toothpick, and the hard part is to try to pull up the hyphae so that you don't clump it together. So you should see nice little strands coming off of your toothpick. And then if I use another toothpick, it makes it a bit easy to sort of spread it out in the water drop. There we go. Look at that. So I'm just spreading out the hyphae as best as I can in the water drop, so it'll be easier to see the structures. There we go. And then I'll take another cover slip, put it on top. Press it down and then blot away the excess water. Voila. Maybe you can see it, it's a little bit unclear, but you might be able to see there's a little bit of a blur inside the cell there, or inside the ring. So there's my fungal hyphae. And let's see what we can find for structures.
Blot away the water. Should it like be dry? Or?
No, because the water winds up being squeezed to the outside of the ring, but the ring still adheres to the cover slip. So you get water that's just on the outside of the ring. So you just want to wipe that away so it doesn't get the microscope, so you don't get the microscope wet.
Yeah. Paper in that.
Yeah. Now that I have the sample there, that. Well, now. See the hyphae for the Rhizoctonia. And depending on what your species is, maybe you can see here, so you get some branched structures forming, but you also should see that there are these little dividing parts in the cell. You see those little black parts? So these cells would be considered septate. So this is a septate hyphae. So all the individual cells have a dividing wall between them. But with some fungal species, they're non-septate. And again, don't ask me which or which when it comes to what species, because I am really not a fungal expert at all. But I do know my hyphal structures. So what I would, and then you can also probably see there's a few other small, I'm not sure, I don't think those are really, those might be spores. I would have to look around, but you can see up here, there are these sort of branched areas. So those, yeah, those are probably spores. So it looks like you're getting some possible conidia formation up here at the top. So you can see these sort of star-like structures, and those are probably the fruiting bodies of this, of the Rhizoctonia, that are going to be producing the spores. So you can explore what your fungal structures look like, and then maybe if you're curious, if you keep track of where it came from, I mean, I know what species mine is, but if you keep track of where your species came from, you might be able to look up what possible species it might be. For example, if it came from the outside of like a type of cheese, for example, that's a specific species that they use. Usually that's a penicillium. Or if it came from bread and it has a green color to the spores that you see, that's probably also another form of penicillium, but it could be a different one, a different species. And you can look at the fungal structures that you see and then compare them with what you see in when you do a bit of research. I don't have a good fungal guide on hand, unfortunately, because I have other guides that I was looking up. But this information is easily available. So
yes, it's a lemon as well. Yeah.
there you go.
Ah,
So you have definitely some hyphae there, and I would say quite a lot of conidia. So being a green mold that grows on fruit with conidia, it's hard to see, because I think that could also be a penicillium. I'm not 100% sure, but because penicillium, when you can see the conidial structures, they usually form these sort of branched, maybe you can't see my cursor. Can you see my cursor? So you'll get these sort of branched structures that kind of look a little bit like a flower, but then that's where you get the fruiting bodies, that's where you get the spores coming from. But here you can see all these different round conidia that are floating around here. And then you probably have a bit more of the fruiting, the conidia forming structures back in here. But you can easily see the hyphae. It's a bit difficult to make out if they're septate or not. Meaning
What's that? Sorry.
Septate. So if you remember from, let's see here, hang on. Sorry, I'll show you what it's usually one of these defining structures. It's one of the defining structures in a fungal hyphae, whether or not the hyphae have essentially dividers between the different cells. So here, for example, you can see if you follow this hyphal strand along, you can see there's this little divider there, and then I think there's one there and one there. That means the hyphae are what's called septate. So septa, you know, a separator, a little membrane separator between the cells. Because some species, the hyphae in some species are septate and some are not. And then actually with some hyphae or some species, you get sort of like a free flow of nuclei through the strand of the hyphae. So you can actually have regions that will have multiple nuclei. So you found that on on a piece of wood out in the forest. This one also looks non-septate. bit hard to make out. Growing on the wood, I would assume it's some sort of saprotroph, probably. Could you see any spores or any coloration on it?
No, it's pretty orangey brown, like the big part of it, but I can't tell anything about spores, no.
So like a cinnamon type color.
Yeah, I'd say.
That could be Serpula lacrimens. So that's one of the what they call the brown rot fungi. The dry rot fungi. Actually, the Serpula lacrimens is a particularly nasty one if you have it in your house. That is huge swamp. That's not a good one. But that one is quite distinct, and that's the thing. That one is distinct because of the color of the spores, which are usually a cinnamony type texture and color. So like an orangey type of dust that's on the outside. And it's particularly indicative if you find it on, obviously, a piece of rotting wood in the forest, that it's most likely of that particular species. But on the page for the instructions, there are actually now, there's 2 guides you can use to try to identify the different species of algae or protozoans. And you can download these, and just to give you an idea of what we're looking at, they're nice illustrated guides to different types of freshwater algae. So you can see some nice pictures identifying what species we have. This is the guide to North American algae, but I think you can probably find similar ones here. So I'm not too concerned about that. So when you're looking through your pond water and also in your columns, you have a taxonomic classification here you can use to try to identify the species that you see. It's quite nice. And I also have a nice one for algae, or rather protozoans. And this one was actually really nice because this actually has some sketches that I thought were quite beautiful. So for looking at freshwater protozoans, so you can see all the different types of structures you may find. So you have flagellates, and you have ciliates and amoebids. Yeah, exactly. Heliozoans, exactly. That's what they're called, heliozoans. And there's a lot of different possible, there's actually quite an extensive collection of drawings here for different species of protozoans that you might find in the pond samples or your columns. So you can really get into detail identifying who it is that you might see. And in addition to the illustrations, there's a really nice taxonomic guide at the bottom. This is actually pretty extensive. So there's a range, you got a lot to choose from. So here you can see the major groups of functional groups of protozoans, and then the major flagellated ones, the amoebids, and other types of amoebids. So feel free to just, if you see something of great interest that you think is just really, really neat, a particular protozoan, just dive in and try to identify it. But the one in the middle. So I think I send you like a square of 4. Exactly. So there's 2 really thick ones and 2 slightly thinner ones. Try the slightly thinner ones there. Start with that and see what you find. Okay. And I will do the same. So here I have my pond water. Okay. There you go. It might be a little too thick. You don't want it too thick, because then it becomes difficult to make out different objects. So just the right amount of sediment, so you get some clear areas in the slide as well. And then a cover slip over the top. Clean up the excess water. And there we have it. No, we don't. I cracked my cover slip. I'm gonna make another one. I think that's the trouble with the thicker ones, is those cover slips are quite easy to break when you start having thicker wells like that. Try this again. There. Okay. Let's see what's growing in the Altoona pond. Or maybe I don't want to know. I don't know. Oh, sorry. I have a very active slide. Let's see if I can get some movies up. If it's a little too thick, that might be a concern. You can start with the thick ones, but if it winds up that you have a hard time focusing on any organisms that are swimming around, you can always switch to one of the thinner rings and you might see them a lot easier. So let me see if I can get a bit of a movie here just to give you an example of what I mean.
I can see little swimming creatures.
That's a good start. Can you make out what they are, shape-wise?
Some of them are a bit round and chunky.
Are they moving fast or slow?
Pretty fast, I'd say. I had a bigger blob that was moving a bit slower. I got a
See if you can capture them on video. This is, if you have the cameras hooked up, I would recommend you use the video in this case.
I have something eating something else.
Oh. Is it a swimmer or a blob?
Looks like a blob. I filmed it, but I'm trying to get a better movie of it instead.
Uh-huh.
See it?
Yeah, yeah. Oh, that's cool. Huh. So it looks like it's shooting out a tendril.
Yeah, I know. I tried to get it. I have a longer video, but it hasn't downloaded on my computer yet. So.
Wow. A blob with a tendril. But that might be that's one of the euglenids, and that's a photosynthetic. But that looked kind of greenish, didn't it? In your video.
Sort of. Yeah, maybe.
Because they're both bacteriovorous, and they can be photosynthetic as well.
Okay.
So that could be the species.